for medical science. He serves as the director of the Arkansas Children's Nutrition Center, a partnership between USDA and ARS, and the Arkansas Children's Research Institute, with a research focus on the role of maternal child nutrition and physical activity in optimizing health and development. He received a bachelor in 1996 in chemistry from Duke University and PhD in 2001 in food science and nutrition from the Ohio State University. Dr. Ferrucci has previously served on the faculty of North Carolina State University between 2016 to 2021, Purdue University, 2004 and 2016. Dr. Ferrucci's research interface of agriculture, food, and nutrition science in the study of micronutrient and phytochemical bioavailability, metabolism, and impact to human health. It has a particular focus on strategies that can be leveraged to improve the nutritional and functional quality of food products for a risk population. Thank you very much, Mario. I remember very well 6 years ago when you visit the Yautepec, and a good time that season. Thank you very much for accepting this invitation, Mario.
Thank you, Arturo. It's funny. I think the last time when I visited, it was when I was leaving Purdue for North Carolina State, and now you have me leaving North Carolina State for Arkansas. Yeah. So maybe the next time will be my next job somewhere else.
Okay, right?
Great. Hopefully not. Hopefully I can stay put for a little bit longer. But thank you. So in speaking to Arturo, I think last time when I was there with you in person, I was able to give a talk a little bit about our research on polyphenols, and really this is a combination of not so much just my research, but maybe broader perspective on the absorption of polyphenols and how not just food but other dietary factors may modify our body's ability to absorb these components, which are bioactive and could be good for health. So just an outline. This is really to talk about factors that are critical. I'll give you a little brief introduction. We'll talk about polyphenol absorption. So hopefully give you an idea why polyphenols are important, but also how they're absorbed. Why the interaction with the gut microbiota, which I'm sure all of you who are students in the area of food science or nutrition, but certainly even from a food science perspective, have heard about looking at foods, designer fibers, and things related to the microbiota. We'll talk a little of food form and matrix and diet. I'll give some examples of some recent research comparing fruit versus fruit juices and how that form may differ. And then the influence of diet and physiological factors, which are also critical to consider. And then finally, I'll end up with something that I think is a new direction, which is where plant genetics are now being considered in terms of factors that could be used to modify polyphenol bioavailability. And what do I mean by that? It's once we know that what is driving better absorption, then we're able to begin to breed for that as a trait. So I'll cover that hopefully today. So first, I mean, berry fruits are the ones that I think, and nuts and other products are so fruit and vegetables in general, but berries and nuts are very rich sources of dietary phenolics. And in this case, we're dealing with multiple classes of phenolics, many of which you may have studied or researched in your own life. So many phenolic acids, hydroxybenzoic acids and hydroxycinnamic acids that you see here. You may have heard in grapes, the stilbene, so resveratrol as being part of the French paradox. But the large class that we deal with most often is the flavonoid class, and they can come in multiple structural forms. And these are components that give the fruits and vegetables color. For example, the anthocyanins here, the blue and red colors and purple colors that we see in the fruits, but also have been shown to be linked to many beneficial effects in humans, cardiovascular benefits by modulating oxidative stress, anti-inflammatory stress, the ability to modify digestibility of foods and reduce glycemic response, which I believe is what I was discussing 5 years ago when I was there in Mexico with you at the time. So it's these compounds, not just the nutritive components of these fruits and vegetables, that are actually quite important. Now, what's also interesting is that some fruits, some nuts, and have very rich source of what we would say are complex, large polymeric forms of these polyphenols. So when we think of things like pomegranate, to a certain extent strawberry, and certainly things like nuts, walnuts being the case, you have these elagitonin compounds. These are very complex, structurally diverse molecules that you see in the skins or in the seeds of these compounds of products that are actually made up of smaller units called ellagic acid. Now, why is this important? Well, these large molecules, just like large polymeric forms of procyanidins, which are really monomeric flavonoids that polymerize in the seeds and skins of things like cranberry or grape, these are not absorbable by the human body. Yet we know that if you consume these large compounds, which were otherwise known as tannins, then we do know that there are health benefits associated with them, right? But we know we can't absorb them. So what's going on, right? So there's an aspect of that as well that adds to the complexity. So in thinking about how these things are absorbed, this is a model that Andrew Nielsen, a former grad student of mine, faculty member at NC State, and I, in 2010, we actually started mapping out, and based on other people's literature as well as our own, how are these compounds in foods being released through digestion and absorbed? And the simple model is that you have bound, this is BPP, bound phenolics in foods like whole grains and certain fruits and vegetables, and you have FPP, or free phenolics. And what do I mean by bound and free? Bound means it's physically tied up in the food matrix, sometimes covalently bound, sometimes non-covalently, but tied up. And it requires a strong amount of digestion to break down and release these compounds through digestion for absorption. So you get primarily that occurs in the stomach and small intestine, and you begin to see some absorption of polyphenols in the upper small intestine, where we see typically absorption of nutrients. There is metabolism that occurs in the intestine. This is phase 1, phase 2, typical toxicological xenobiotic metabolism, which means the body treats polyphenols like a compound that's toxic. So it metabolizes it by conjugating it and preparing it for excretion. And some of that is to push it back into the intestine. Some of that is to push it into absorption and further metabolism prior to excretion to urine. But in that circle, it can see other tissues. It can get into sites that are associated with health benefits. So you might say, why would the body do that? If it's good for you, why doesn't it try to keep it around? Well, these tend to be water-soluble compounds. They tend to be obscure and foreign to the body. So the body tends to work to clear compounds that don't have a strict nutritional purpose or are at concentrations beyond traditional nutritional purposes. So it's viewing it very much like it does a toxic compound, even though there's no real evidence that many of these compounds are very toxic. This process is very inefficient. You may only be absorbing 1 or 2%. So a lot of it makes it into the lower intestine, where the microbiota will then further ferment and break these compounds down into colonic metabolites, which are also the primary metabolites that we see. So the bacterial metabolites are the ones that, in fact, are likely to be the primary ones we see. And so over the years, most of us have focused on studying the upper GI. And these include factors that alter this digestive release, disintegration of the food. So whether it's breaking down starch and protein, lipid, all that interact with polyphenols, food source, matrix, the amount of processing makes food more or less digestible. And then interactions, as I mentioned, the ability of polyphenols to interact with protein and carbohydrate and fiber can tie these up in the gut. And then there's other interactions as well. More recently, there's been a significant focus on what is altering the polyphenol digestibility in the lower gut. So this includes composition of digestible and fermentable fiber, as well as phenolic-phenolic interactions that lead to microbial interactions and fermentation. And I'll speak a little bit to that next, which is the paradigm from when I was working in this 10 years ago to now has been that these polyphenols derive their health benefits directly. But what we do know is that they come from foods, and they come from foods, and in those foods, they tend to have structure. They tend to have components that they physically interact with: carbohydrates, fibers, protein, lipids. Combined, these, as you consume, will affect what? The gut microbiota. And you can alter the gut microbiota depending on what that food composition and diet are. And it could be good or bad, right? What we do know is that there is an effect, as I mentioned, metabolism by the body, metabolism by the microbiota, and health effects can be mediated either directly or indirectly. In other words, directly by the polyphenols and their metabolites, and also by the polyphenol interactions to the microbiota. Now, what's also interesting, as I mentioned, the colonic metabolites or the microbial metabolites. We know that microbiota generate things that are important for the body: short chain fatty acids, branched chain amino acids. One of those is phenolic metabolites. These are the postbiotics that we think now are critically important to tell the story of why fruits, vegetables, and botanicals may in fact be beneficial for human health. So the fact is, it's not so much what we consume on this side. It's actually what's being formed over here, as well as over here, that is driving the health effect. So there's a disconnect between what's in the food versus what's in the body. And it's important to understand that relationship with the microbiota. Now, this can get very complex. And if you think about, like, here are 3 flavonoids and phenolic acids: chlorogenic acid, common in coffee; hesperidin, common in citrus; and then flavanthriols, monomers and polymers, common in cocoa, for example, or tea. The gut microbiota will take those and break them down. So you'll get ester hydrolysis and release out the phenolic acid. You'll get C-ring fusion, so it'll break right down the middle here or here, and also generate similarly phenolic acids, because if you think about how flavonoids are made, it's actually a fusion of 2 phenolic acids. At the end of the day, you basically condense down to a group of 5 or 6 classes of phenolic metabolites, and then they can be conjugated by the body's own systems: glucuronidation, methylation, and sulfonation. And so even though you may consume one molecule, flavanthriol, let's say you only consume cocoa, you're primarily flavanthriols, you're going to get a mixture of these compounds in the blood. So you go from 1 to maybe 30 compounds. How does that look? It looks like a subway system. So this is from my colleague, Colin Kay, in North Carolina State. Colin's an expert in the metabolism of these compounds, and he shows here structurally how cyanidin-3 glucoside, this is the anthocyanin pigment that provides a lot of red color, or even purple color, I should say, to corn, purple corn, okay, and blue corn. So you're able to kind of see this one compound can break down to cyanidin, then it can break down to caffeic acid, and then all the other metabolites. And you can see isoferulic all the way down until you get to hippuric acid and phenol. But in the meantime, in the blood, you'll see a combination of all of these compounds that is derived from one. And that flux is driven by both human and microbial systems. So again, how do we unwind what's important to think about in terms of absorption and effect? Keep in mind some simple things. If you go back to what I provided earlier, you can quickly see, hey, there's low metabolites from microbials in the small intestine and a high abundance in the large, whereas here in the upper GI, you have a high abundance of native compounds and a very low abundance of them in the large intestine. So that means you typically have 2 points of absorption. So if you were following absorption of polyphenols, what would you see? Well, you'd see typical absorption with a half-life, 2 hours. This would be the peak of those flavonoids, right? These are those smaller molecular weight gut microbial metabolites. So what are we talking about? A much larger wave of compounds being absorbed. It's probably even larger than this, but look at the time. If you consume eat your fruit or cocoa, you're going to absorb some things very early. Then the microbes will get a hold of it and you're going to absorb a lot more over 24 to 48 hours. Okay. So the response carries on because it takes a while for our bacteria to work. And so in that space, a lot of factors can alter how much or how little you absorb and the form in which they take. So one of those, these are factors that if you went in the literature, you would see. Food matrix factors are key. So processing, macronutrient, micronutrient, and then the form of the food are all important because they impact bioaccessibility, which is the ability of these compounds to be released from the gut and made available for either absorption or metabolism by the gut microbiota. And if I were to summarize it, you would see effects of solid versus liquid foods. You'll see food or supplements. So if you take away the food matrix and you only have the flavonoids, you're going to see a big difference in the rate of absorption. You'll look at these interactions, as I mentioned before, protein, polysaccharides, micronutrients. There's lots of great papers. We've written reviews, as have others. I would encourage you to read these, and I'll certainly send the slides for this presentation afterwards so you have these references. But then the type and extent of processing, juicing, homogenization, etc. Now, I'm going to give you some examples on these today, the food, supplement, and juicing, as what I would say are things that consumers see frequently that actually impact the amount that could be absorbed, but also impact the type of metabolism you might see. Okay, so I'll talk about those today, but I just wanted to highlight interactions with macronutrients are key, and they probably drive some of those effects that we see on processing and on formulation. If I had to tell you about polyphenols, they actually interact with everything. We spend a lot of time working on carbohydrates, but the most well-known, probably known to all of you, is the interaction of polyphenols with protein. And you probably know it from drinking tea or having a bitter or astringent fruit at some point in your mouth. And it's because free polyphenols have the ability to interact with salivary proteins, and they contribute to the astringent reaction in your mouth. So if you've had black tea or green tea, you notice a drying, puckering in your mouth. And that is the polyphenols binding to salivary proteins, causing them to compact and polymerize, and then ultimately fall out of solution in your mouth. And that is exactly it. Or if you had a nice bottle of red wine that has a high tannin content, and it relates to these large polymeric compounds that bind to protein. And of course, what's the solution? Well, you can add milk, not to the wine, but to the tea, right? If you add milk to the tea, what are you doing? You're pre-complexing these polyphenols to dairy protein, and you don't taste the astringency. And then the dairy proteins digest it, and it releases the phenolics in the gut, and they become absorbed. So these interactions are important. So it becomes critical if someone is going to study the effects of a fruit or a vegetable or any kind of food to understand if we remove that matrix, do we alter the behavior in the mouth and in the gut, and do we alter absorption? And that's actually one of the questions we had. Was working on a project with Connie Weaver's group at Purdue, and they were interested in studying blueberries and studying blueberries in the context of bone health. And I'll get back to some other learnings we had from the study. But in blueberries, you have two classes of polyphenols: chlorogenic acids, same as you see in coffee, and flavonoids, primarily anthocyanins and flavonones. So part of the study was to look at very high dosage levels that are not achievable by diet. So how do you do that? Well, you work with extracts. And so we were fortunate enough to have native fruit and an extract. This is not the brand we use, but to compare, an extract that is made from that same fruit. And we were able to do this as part of a toxicity study where we were doing screening to find out if we could go too high on concentrations. And so we compared a freeze-dried blueberry that had all of the food matrix to a blueberry extract that had none of the food matrix. This was a hydroalcoholic extract of pure polyphenols, pretty much. And we matched doses, and then incrementally went up in dose 20 times. Okay, so this is from multiple serving, maybe 2.5 servings of blueberries versus something like 20 some servings of blueberries. So based on what we wanted to follow, we could follow the flavonoid absorptions, the phenolic absorptions, but we could also follow the main classes of microbial metabolites. cinnamic acid, phenylpropionic acid, phenylacetic acid, benzoic acids, hippuric acids, benzaldehydes, and then phase 2 metabolites, which are the conjugated versions. So here's what the data look like. And what you'll see here is we fed over 90 days. This is day 7 and each day of analysis over that time. You'll see individual metabolite classes are here. So this heat map is what that is. But these are the overall classes, the native flavonoids, the host metabolites, and then the microbial metabolism and host metabolism all the way to hippuric acids. And what did we see? Blueberry fruit, you see a very low level of phenolic metabolites and that change over 90 days, but you start seeing an increase over time of hippuric acid metabolites. If you take away the matrix, what do you see? You see a little bit of a backup to some of these other phenolic acids that are microbial, but there's a slower proportion getting to hippuric acid. So immediately it tells us that there's some metabolism difference, but for the most part, fruit and extract, similar levels, similar response. As you go up in levels, though, what do you see? Higher and higher levels further back. So this is more what's in the fruit and the early stages of metabolism than the end. So you see higher and higher levels as you get all the way back here to, in fact, compounds that are being less metabolized. So I don't show it here, but the point is you see big changes in the microbial communities and shifts in the microbiota where you begin to lose diversity at very high levels. And so too much, not good. You begin to unbalance a bit of what's happening in the gut. But it also means that it's very difficult to predict that any benefits you see from food will be the same benefits you see from high doses of extracts, because you'll see different compounds in the blood, different compounds making it to tissue. So not quite the same. So we have studied for years, tried to understand why this is happening. And we've relied extensively over that time frame the use of an in vitro digestion model, which is somewhat important for us to really define what I would say are the matrix effects. So we can compartmentalize what happens in the stomach, in the oral compartment, in the intestine, and ultimately identify compounds that are absorbable and then be able to relate those to the amount that's in the food. We've leveraged this model to study matrix interactions in tea and cocoa and fruit, solid, liquid, with and without micronutrients. We've looked at food forms. So if you make foams and gels out of polyphenols, how does it change the absorption? We've also studied whole grains because we're really interested in understanding how they interact with starch and fiber. So these models have helped us really get at many of those food matrix questions that I was mentioning before, right? A lot of those. So how about us using models like this to better understand food form? And so one of the questions that came to mind, and this is particular to the United States, but it's also important as we go to Latin America, because fruit and fruit juice consumption is such a cultural and important part of our everyday diet. And in the US, there's very big concern that fruit juice is going to be removed, especially 100% juices will be removed from dietary guidance out of fear for high levels of sugar. Now, to be clear, there are very little differences between sugar that's in the grape and in the fruit. The concern is really the potential for overconsumption of juice, okay? So juice, however, and fruit is a number one source of these flavonoids in our diet. So if we are looking at a type of consumption, do we need to consider how fruit and juice may differ in their ability to deliver on polyphenols? And just to give an example, where are most of these polyphenols located? When we're talking about fruit, this is Concord and this is Niagara. These are two Labrusca grape varieties, very common in the US for juice grapes. And you'll get a diverse array of flavonoids, anthocyanins for color, flavanthriols, procyanins, flavonols, and phenolic acids. And they're almost all concentrated in skins and seeds, very little in the pulp, okay? So the key thing is you may consume grapes and you'll consume the skin, but most people are going to spit the seeds out. And the seeds, if you go to a light colored grape, are 95% of the flavanthriols. So you're going to lose a lot of the phenolics if you're eating fruit. Interesting thing is when you process in the juice, what do? you're actually extracting that fruit. So you may not extract everything, but you're using enzymatic techniques. You have pressure, you have heat, you have thermal processing as well in pasteurization. You're creating an extract of fruit that is standardized as 100% juice. It has high shelf life, and it also critically has perhaps compounds from seeds or components that would not be consumed extracted into it. So how do these differ? And, allegorically, I just want to point out, if you look in the literature, the majority of the health benefits, for example, for something like grape, have been shown from 100% juice consumption, not fruit. Most people study juice, could be for convenience and other factors, but do these benefits extend and are they the same? So we actually have this model, as I mentioned, and we actually did something interesting. We coupled simulated mastication. So here on the left, you can see human masticated grapes, and on the right is our simulated ones. Why did we have a simulated? Because we had hundreds and hundreds of samples to process, and my graduate students at the time had no interest in chewing everything. So we came up with a way to simulate mastication in oral phase. We had matched juices to the grapes, passing through our 3-stage in vitro digestion, create a digesta that then could be screened for intestinal absorption using CACO2, a human intestinal cell culture model. But then we did something different. That's our normal approach. But then we would take the non-digestible fraction and ferment it in an anaerobic fermenter with open biome source microbiota, like fecal inoculum, create fermenta, which could then also be measured for absorption. Now, why is that unique? Because fruit and juice are different. Our hypothesis was really that juice would drive a lot of absorption in the small intestine, and a lot of the effects would be rapid. Whereas with the fruit, with all the matrix around it, you would need the digestion from the large intestine to release many of the phenolics that are bound in the seeds and the skin, okay? So that was our hypothesis. This is Zulfikar Mohammed Shah who did this work, and I'll show you a little bit of the results. So just to highlight a few of the results here, what you have up top is content. And in deep purple, you have the grape for the Concord grape, and deep green, you have the grape for the Niagara grape, and then the lighter colors are the juice. So when you look at anthocyanin content, you see that juices are lower than fruit. Again, we're not 100% extracting when we make juice. Similarly, you see phenolic acids are a bit higher for the juice as they are here in the Niagara grape. Again, raw material. Why is it so high in phenolic acids? Enzymatic treatment is releasing phenolics that are bound with the fiber. So again, similar profiles for white grape. Grapes are better than juice on the dark grape. But here's the bioaccessibility. What did we find? In almost every compound class, you see a significant higher level of bioaccessibility from the juice. And this is absolute, so milligrams per 100 grams of product. And that is driven, same thing with the green grapes, by the high level of relative extractability from that fruit. So when you look at anthocyanins, your body chewing it in digestion has a very hard time releasing flavanthriols and anthocyanins, but the juice, it's already pre-extracted, so they're very highly bioaccessible. And so you see that reflected in the fact that juice becomes a better source, a more bioaccessible source in the small intestine than does whole fruit. If you look now at absorption at the small intestine, this is the CACO2 data. What do we find? We find that juice, for the most part, this is flavanols, anthocyanins, phenylacetic acids, etc. But the primary compounds are the anthocyanins and flavanols. So just focus on these two. These others are actually chemical breakdown products. What you see is that the anthocyanins and the flavanols are much, much higher out of the juice than the grape. So again, these structures that are in the fruit must maintain, and they don't release for absorption by the intestine, even though they're soluble. Now, these are all chemical degradation products, and even those you tend to see a little bit higher from the juice than the fruit in kinetics of transport. All that would combine to tell us that if you consume juice versus fruit, you're going to get more into your blood in the first 2 hours from the juice. But what happens after? So here's what happens once we start moving into the fermentation. This is native fermentation of phenolics. So what we see is Concord grape, Concord juice, and this is a control, which is without fecal inoculum. So sorry, this is off a little bit, but what you'll quickly find out is that we're seeing, in fact, loss of the native compounds very similarly between juice and Concord grape, albeit much higher concentration in the Concord grape. Why? Because it was not made absorbable. So we've already absorbed out what's in the juice, and now you're putting more of the fermentation substrates into the inoculum, so more to generate more microbial metabolites. And this is what you begin to see here. Almost 2 to 5 times higher, 3-hydroxyphenylpropionic acid, 20 versus 6 from the juice. Very important point. We're just seeing far higher levels forming of certain metabolites while others don't change. But juice and grape are forming similar patterns, but at different rates. And in fact, you become more bioaccessible from fruit out of the fermentation because there's more substrate. From the transport perspective, though, there's no difference. They all behave about the same, as you see here. There's some subtle effects on some of them where the grape is a little bit better than the fruit, but and so you're getting into the situation where, well, whole fruit may be better in the lower gut than the juice, which is better in the upper gut. Big summary on this looks like this. If we were to look at total bioaccessible phenolics versus cumulative transport, which is actually what's in the blood, what you quickly see is that small intestinal juice is the main driver of what goes into the blood, whereas you catch up quite quickly in the colonic area if you're fruit. So all in all, though, you're all about similar levels here. There's a little bit lower juice here, but the cumulative amount that's making into the blood is the same. It's just being absorbed in different places. So whole fruit versus juice, if you need an acute response, juice is better. If you need something over a day or two, the fruit is better. This goes to tell me these are far more similar than different. And unfortunately, policy people want to make juice bad and fruit good, but they actually are behaving more similarly than we think. Okay, change of pace. Talk about diet and physiological factors. Health status, the ability of the body to adapt, and then the gut microbial communities. We learned early on that if you feed polyphenols to people that have not seen polyphenols often or have different health status, they're lean versus obese, you might get different responses. This is just an example from a study with Janet Novotny's group at USDA ARS. A pilot study of 12 people, 6 and 6, healthy, and then risk for metabolic syndrome. The big difference here, obesity, right? So you had a BMI of 30 versus 23. We fed a lot of polyphenols, resveratrol, grapeseed extract, and Concord grape juice. And we did a pharmacokinetic assessment of bioavailability on day 0 and day 10. And I just wanted to show you a little bit of what that looks like just from 2 compounds. Here's epicatechin. This is the lean, healthy group, okay? BMI 23. Day 1, this is the absorption of the polyphenols. This is day 11. They absorbed more. Now, why is that? I'll tell you in a second, but they absorbed more. Here's what happens with the obese, though. Day 1, day 11. They did not adapt. They did not absorb more. They absorbed overall less. So if you're healthy versus unhealthy, you may react differently to polyphenol treatment. And in parallel to this, we were doing studies in animals tracking the microbial breakdown of these compounds. And lo and behold, here's catechin and epicatechin, and this is over 10 days in animals. And what do you see? You see release of epicatechin and catechin, the monomeric units from what? From the large polymeric grapeseed extract. So over time, the microbiota becomes better at metabolizing large compounds. So all of you that have studied fermentation perhaps understand that there's a lag phase for the bacteria to get used to this. But they adapt to a point where they can generate and survive and thrive in that environment by breaking down compounds like these large polymeric components. And so that's probably driving the increase in bioavailability that we see. So again, it's hand in hand. What's in the product, but also what's in the gut is going to drive the response and perhaps drive your ability to adapt to it. Lean people may have a more adaptable, amenable gut microbiota than obese individuals and not healthy. And this is an idea that has really come up in a lot of research, this notion of metabotypes. So it's not just eat healthy, but do you have the right gut bacteria to generate the right compounds that are in fact reflective and responsive to health? So I mentioned the elagitannins before. There's very good literature. This is Paco Barberon's work out of Spain, where he's looked intently at the breakdown of elagitannins to ellagic acid, but more importantly, to the compounds called urolithins. This is a specific type of metabolite, Gordonobacter, which is one of the driving bacteria that drive that. And you can have 3 different kinds of metabotypes. You could be a metabotype 0, a metabotype A, or a metabotype B, depending on the end stage urolithin you make, if you make urolithins A or B. Now, why is that important? Well, it turns out, and these are some of the data, and I'm not going to take you through all the data, but it turns out that if you do a clinical trial and you look at all the individuals, this is a clinical trial with pomegranate looking at cardiovascular risk biomarkers and primarily looking at serum lipids. You don't see any effect. There's no effect of pomegranate treatment. But then when you break up participants as A or B metabolizers, you see that the B metabolizers have significant improvements in cardiovascular endpoints. So again, some people respond, some people do not. The notion is now you can identify who those responders are and then move forward, okay? So understand the food, understand the microbiota, and understand the individual. You may be able to piece these together and determine the effects. Now, these notions all came together for us several years ago when we had a fairly large NIH project that looked at linking specific metabolites of blueberries that we would define through traditional pharmacokinetic assessment that linked up and activated, these metabolites would activate NRF2 pathways, which are the antioxidant response pathway, anti-inflammatory pathways that would have outcomes on bone. This is a very large NIH grant, and we were to study blueberries. So as you can see, I was on the team with Marianne Lila at NC State and Connie Weaver at IU and Teresita Bolito, who's actually now here at University of Arkansas as well. Now, I'm a food science and an ag person, so I'm sure many of you are similar. So when someone says, I want to study a blueberry, or I want to study mango, or I want to study papaya, or I want to study apples, the first question is, what do you mean by blueberry or apples or whatever? Because there's hundreds of varieties, and they may not all generate the same compounds. And everything I've told you about the matrix and everything I've told you about the people is, what do you mean by blueberry, and who do you mean by people? And is that something I can map out? And thinking about this from the standpoint of blueberries, what's really interesting is there's a lot of variability in the polyphenol content within the broad collection of Vaccinium species. So when you say blueberries in the United States and in Europe, we get thousands of different genotypes, okay, of blueberries. And some could be literally no color at all to super, super high in polyphenols. Well, how do you begin to choose what's relevant to study? Because when you go to the store, you don't have a single genotype. It's usually a mix. So the research that we're getting ready to do, is the research we're getting ready to do relevant to consumers? And so luckily we had data from Marianne's group on 267 genotypes. It was over 1,000 plants. And we had anthocyanin data in tabular form. And I'm going to show you now the piece of data that we have never published that is my favorite piece of data. So here's the 267 varieties on principal component plot to show variation in anthocyanin content. And if you look at that, and I would ask you as students who study statistics or faculty who do statistics, what's your first thought? Your first thought is, hopefully, that's a lot of variability. In other words, it's evenly spread variation too, which is great if you're a breeder. It's not what we're taught if you're a nutrition scientist or a food scientist. You want tight data that's reproducible. But this variation is important because now it allows us to make selections to screen for things that allow us to account for all the variability that consumers might see. So what we did is we set up these coordinates on this plot, and you see them in red. And we selected against the second principal component primarily, relevant cultivars or varieties that we could screen for bioavailability, just to ask the question, are they different? And here's what we found. We actually, in blue, you see the content of the blueberry polyphenol. So you can see we had several that were very high, Ira and Onslow, and then a breeding cross, and then a wild blueberry, which is a low bush blueberry, very high in polyphenols. And then you had one that was very low, Montgomery. And then you had a priority selection. What is that? That was the actual cumulative blood metabolite levels over 8 hours, okay, of the polyphenols. And so the one that had the lowest amount of polyphenols had what? The highest blood levels of polyphenols. And actually Montgomery is right out there. There it is. Very different than you can see than these. So it made us think, if someone were to ask you, which berry do you want to study? You probably would have picked one with the highest amount, but then you wouldn't have seen the blood response. Now, why is that important? Fast forward 4 years later, after we selected Ira and Montgomery. So we selected these two to study: high, low response, low, high response. And I'm not going to take you through the science of this, but this was on skeletal protection and promotion of microbial diversity. And Amy Sato and Teresita Bolito, 3 blueberry genotypes: Montgomery, Ira, and then the wild blueberry, which is always studied as being super healthy. Out of those 3, remember, Montgomery is low, Ira and wild are high. Only the Montgomery was protective in OVX female mice for bone loss. It was also associated with endogenous antioxidant response and increase in gut bacterial communities, diversity. So it was lower in phenolics. It interacted with the gut bacteria and generated higher levels in the blood and a positive physiological response on bone. The others did not. This is important if you're studying, because now you can say we might need to understand the basis of why there is a response. And this became another project for us. So when you study berries as food scientists, we study them for quality traits: skin thickness, size, fiber, pH, sugar, and polyphenol profile, because we're interested in color. Those are driven by the plant's genetics, and also driven by what? Where it's grown and how, the environment. It can also be modified by processing. You can lose color, gain color, etc. The question is whether any of those individually, the genetics per se, or the processing, can impact the bioavailability or the bioactivity. And so luckily I'd been working with Massimo Iorizzo, great colleague at NC State, who was already studying and creating a framework to study anthocyanins, and in particular, acylated anthocyanins. And that just means anthocyanin glycosides that have phenolic molecules that acylate the sugar moieties, to really understand the genetics of that process. And he had done so and identified the genetic targets and the enzymes responsible for acylation. And it's important because acylation, we know, alters bioavailability and physiological response. So the first question we had was, hey, is Montgomery a highly acylated anthocyanin? And the answer is yes. So why is that important? Well, when you measure an acylated anthocyanin, you're measuring one compound, right? But it could be that you're measuring one compound that breaks down into 1, 2, 3, 4, 5 different phenolic acids. So you're releasing much more in the lower gut, which could be why we're seeing a higher plasma response from that variety. So the question again was leveraging the same models that we did before. You know, how do we now screen this variation across a germplasm collection of 276 or 1,000 different genotypes if we're trying to study bioavailability as a trait? And so the original in vitro digestion is quite cumbersome. We can do maybe 30 samples a day, a lot of manual pipetting. So we spent the time with a robot, created a high throughput system that allows us to do 96 at a time, almost 300 a day. And now we can do digestion of fruit and begin to fingerprint and phenotype bioavailability to do what? The same type of genetic studies that were done on content can now be done with a phenotype of bioavailability. So we can begin to identify the traits long before we select these fruits for study. We just got lucky selecting Montgomery. We want to help people get there without luck, but with data. So we've published a paper with Massimo in 2020. And in that paper, it highlights the 2 years of data where we did this screening of bioaccessibility on 180 accessions. Just to quickly highlight it, this is the content right here. And you can see phenolic acids and anthocyanins year on year, 17 and 18, very similar contents. Similarly for flavanthriols and flavanthriols. But if you look at the relative bioaccessibility, this is the percent that is bioaccessible in our high throughput model, you quickly see, again, a broad range of variation. There's variation here, and there's difference year to year. So the key thing is that we're interested in is seeing this wide range of variation in this box and whisker plot. So again, from a low of 10 to a high of 70 something, and in this case, the opposite, where you even have some over 100. But the point is we want variability because variability allows us to identify genetic factors. And again, absolute bioaccessibility, also high variation and some consistency year to year. I know the time is short, so I'll skip through that and I'll go to this. These are the individual anthocyanins, the pigments from blueberry. And you can see in 2017, high variability with several outliers up here that are very high for cyanidin. And again, broad variation across individuals. There's some consistency with 2018, just with cyanidin being quite a bit higher. It seems like these outliers kind of began to dominate. Maybe you had more that went up there, but the point is you can actually capture all of the fact that there's a lot of variation, but consistent across this group of 180, 120 different genetically distinct berries. Acylated versus non-acylated. So what did we notice? Acylated are, in fact, far more bioaccessible. There's still variation, far more bioaccessible, and it's very consistent year to year. That's an important finding. Now, when you start correlating these things, it's very difficult to find strong correlations just by saying, hey, let's look at 2017 and 2018. More importantly, there's some connection primarily between high correlation between absolute bioaccessibility, which is an amount, more so than relative, which is a percentage to the content. That being said, you're able to plot out relative versus absolute bioaccessibility and find individual berry genotypes that may be, interesting to study or to put into biological testing because they have both high relative bioaccessibility, but also high total. That would be like gauging Montgomery, and that is probably one of those that's sitting in here, okay? Now, the next thing that you can do when you work with a geneticist is begin the process of understanding, is this a breedable trait? And they do that by determining broad sense heritability, which is the amount of variation on this trait, whether it's concentration in pink versus relative bioaccessibility in blue and versus absolute in green. Now, Massimo, I had already told you, had done the studies to show that breeding has high heritable trait is under genetic control. So it's not surprising that pink is quite high, and anything over 40 or 60 is considered to be moderate to high. Okay, once you get over 60, you're high sense of heritability, which means it's under genetic control. What we see under relative, though, is there's several individual ones, non-acylated anthocyanins. You look at individual anthocyanins, there's anthocyanin glucosides, several of these, delphinidin-3 glucosides, cyanidin-3 glucosides, where we're passing the 60% threshold, telling you that there is genetic control of the relative bioaccessibility, and that, of course, then drives the green ones where you see quite a high level there. All in, this tells us that bioavailability itself may be something we can control through breeding. We don't know the genes yet. That's what the QTL analysis is going to tell us. It could be something related to texture. It could be something related to what's bound and not. It could be something related to profile. The point is that there are likely certain gene clusters that drive this phenotypic effect, just like it would drive sweetness, color, and other breedable traits. So is this something we can move forward with in the future? So I'll stop there. I just want to give a summary. So first of all, food form and formulation can impact polyphenol bioaccessibility. It's critical to consider the distribution of the complex non-absorbable polyphenols. These are these big procyanidins, big elagitanins, because they are going to interact with the bacteria and drive many differences we see in response. I showed an example of how diet, dose, and length of exposure all alter absorption. So when you're thinking about designing products or you're thinking about specific health applications, it's very important to consider all of those things. As is it important to consider nutritional health status. If you're making products for diabetics or you're making products for other at-risk people, then you need to consider that they may not react the same way as healthy people do. But the last part is the one I really want you to keep in mind, is that the plant genetics may be leveraged themselves to help us better breed fruits that are actually better at delivering these compounds and the benefits associated with those compounds. So I will stop there, and I just want to acknowledge Teresita Bolito from UAMS. So she's left IU and is here and done all the work on bone. The good colleagues at NC State that I still work with, Massimo Yorizzo, Colin Kay, and Marianne Lila. And then, of course, Connie Weaver and Cindy Nakatsu at Purdue. Connie is actually now at San Diego State. Howie DeBello did a lot of the work up as a postdoc. And then the great graduate students that I've had over the years at NC State that worked on the projects that I specifically showed you. Maria Mines, who was Connie Weaver's student, Zulfikar, Haley, and Dennis, who worked with me. Dennis also worked with Connie. And then Sydney Corbin and Candace Nunn, who are great technicians at NC State that were just the masters at managing a complex project with thousands of analyses. So funding, of course, from Foundation for Food and Ag Research and NIH, and I will stop there, knowing that maybe we have some time for questions. Okay, thank you.
Oh, thank you. Thank you very much, Mario, for your excellent talk. It's very interesting. In this moment, we had a time for somebody had a question. Here is present the Dr. Ariana that is support the translation. Up to you if some attempts require a translation of the question. Feel free to send.
I will do my best in Spanish.
Spanish. Okay, Mario, great.
Well, my Spanish is not that great now. In Italian would be fine.
Okay, great. Thank you. Some question, please, in the chat. No in this moment, but the people, somebody can open the microphone and to give the question directly, please.
Arturo, I'll send the slides.
Okay.
I have a question, Dr. Peruzzi.
Yes.
Thank you so much. I learned a lot from your presentation. For example, I will know throughout the seeds of the grape since today. Well, I'm not an expert in this subject, so I have this question. Colonic fermentation prefers polyphenolic component than resistant starch or protein resistant?
So let me understand, you're asking if the microbiota would prefer to digest polyphenols versus resistant starch, let's say.
Yes, exactly.
Well, I think it's an issue of survival. Many of these polyphenols are toxic to some bacteria. This is why the plant produces them, right? In some cases. So if you have a high level, it's probably in the best interests of the community to have some bacteria that can break down the polyphenols, but there's not a lot of evidence that they use the phenolics for energy. I think there are some that do, but whereas it's using resistant starch, it's fermenting it for energy. And it's creating short chain fatty acids and other things that the tissues then use for energy. So I think it's a little bit of both. I think that there isn't necessarily there's not a preference until it realizes that if it doesn't digest it, it could be very bad for the community. And to be clear, it's never one bacteria. It's typically probably multiple, it's probably community structure that's important. I'd refer you to Steve Lindeman. I don't know, Arturo, if you've had Steve talk. Yeah, but at the Whistler Center, he studies, there's these community relationships with starch structure intently. And I think with the polyphenols mixed in, it's a potentially toxic to the bacteria. So they have to defend themselves by digesting it, perhaps over going after energy first. Depends on the dose, but it's a good question.
Okay, thank you. And another one, there are studies that show they produce a specific short chain fatty acid in the gut microbiota from polyphenolics.
Mm-hmm. Are there studies that show that?
Yeah.
Not so much. I think that there are studies that show if you provide fruit or phenolic extracts, you see increases in short chain fatty acid production. But I'm not sure I would say it's derived directly from the polyphenols. It's perhaps acting in a prebiotic sense, in the sense that it's promoting community structures that might be generating short chain fatty acids from other substrates. But I'd have to go into the literature to be sure, but I don't believe that there have been a significant amount of research to show that the phenolics themselves can be used as a substrate. But there are papers that show that you see enhancement in short chain fatty acid production, specifically butyrate, with phenolic rich diets. And again, that could be like what Teresita Bolito found, which is you're really just promoting alpha and beta diversity. So you're actually showing an enrichment of the right type of bacteria in a very prebiotic sense, but not necessarily as directly as substrate. So, I used to argue with the folks in the Whistler Center all the time. So Arturo may remember this, but when I was at Purdue, they would argue that phenolics shouldn't be part of the Whistler Center because they weren't fiber or carbohydrate. And I said, their formula is CHO. So they are carbohydrates chemically. I said, oh, okay. And they're like, well, they're not fiber and they don't act like soluble fiber. And I said, well, they promote certain bacterial community structure. So you have a CHO molecule that can be fermented by bacteria and alters structure. Boy, it sounds like fiber to me. So probably doesn't fit the strict definition, but it could be contributing to what you see in effects on short chain fatty acids for sure.
Okay, thank you so much, Dr. Ferrucci.
We have something in the chat. When we speak about interactions with starch polyphenols, between starch and polyphenols, which is the main responsible for the formation of these complexes? Yeah, so it sounds like some of the work we've done in this space, so I haven't presented it here, but we have worked on creating complexes purposefully between starch and phenolics. And what we think is happening is V-amylose type complexes. And we know that in some cases, it's going to be very dependent on the nature of the starch and the nature of the phenolic. We have not systematically studied that, but there are two camps. And it's good that you're thinking, because you put down in this molecular weight and monomers, right? So if you follow Joseph Awika's work at Texas A&M, Joseph is focused more on large molecular weight phenolics. I don't think those are complexing as much as what I would say interacting and creating large, what I would say, structures around the starch granule or starch through hydrophobic interaction or even hydrogen bonding. But it's a different kind of that's not really a complex. It's an interaction that's slowing digestibility of the starch, but these compounds are so large that they can't fit in any structure like you would see. We found, for example, when you have potato starch, which has a fairly large core, you can get small molecular weight phenolics into that core. You could load it very effectively. We've also seen evidence that maybe larger flavonoids can, but not polymers. So we study two different things. Our group is focused mostly on characterization. I'm happy to send you some of those papers if you're interested, of that small molecular weight structure and non-covalent interactions. Fairly rigorously has studied that already. There are effects, and they are dependent both on phenolic and on starch, and we can make resistant starch in that process. But there is definitely an effect of the larger molecular weight that's driven by other factors that I have not seen good chemistry on. And it's not a critique of the individual investigators. It's just, it's a lot harder to study when you don't know the structure of the large compounds. So, you know, they're at a chemical disadvantage. They can't get the structures, but that's a very good question. Okay, we got a couple more. Do we know about the polyphenol bioaccessibility, diversity, and quantity when we drink ferments like kombucha and others containing berries? That's a really good point. There's a really growing tendency to think about if we can preform the postbiotics by fermentation. Now, remember, primarily what we see in the lower gut's driven by anaerobic fermentation, or at least to a certain extent, facultative anaerobes that are functioning in low oxygen tension. That could be something to consider. It's just a little bit riskier to do that type of process with food for food safety reasons alone when you're dealing with botanicals that could have a lot of spore formers, right? We wouldn't want to be making bot toxins out of them, although the pH should be able to control that. But there are studies that actually on fermented foods are showing that some of the postbiotics are being driven through those interactions. So that's something to scan the literature for. I would be really curious if we would be able to match it. The problem goes back to matching the correct postbiotic for the right people. So imagine, if you will, this metabotype piece. We assume that if you make urolithin B, you're going to have cardiovascular benefits. And if you make urolithin A, you're not. Okay. So what if I just feed people that have urolithin A metabolites, but urolithin B? Why not just feed it to them then? The odds are you may not see the benefit. This has been shown in equal from soy. You can take pure equal, which is a metabolite of a flavonoid in soy, and equal producers get bone health benefits. Non-equal producers don't. You can put a lot of equal in and you won't see the benefits. So that tells you that that relationship is important with the bacteria in the gut as well. So it can't just be dimensionally driven by the metabolite. And to a certain extent, it tells us the metabolite may be more of a marker than an active compound in this case. So if you're going to go about it with the food solution, you have to be sure that the compounds themselves are relevant. So then it takes a little bit of study. That's a loaded question, the next one. In clinical trials, what is the minimum size you suggest to study a functional food? Well, the answer is real simple. It depends on what your endpoint is. If you're studying bioavailability and you want to compare it between groups, you don't need a large sample size, especially if you know the variation of the response you're getting. We were able to see statistical significance with N of 12. Typically, we would probably look at an N of 12 to N of 20, depending on our bioavailability study. But if you're studying blood pressure, or you're studying neurocognitive benefits or vasodilation, you're probably looking at having to calculate the power of that study based on the variation in that population. So you would either have it from literature, or you would do a pilot study to understand the variability of the response of your marker. And then you would do a power calculation, try to be at about 80% power. So it would not be uncommon to see trials with 30, 40, 50 people, if not more. Some of the studies we have here in Arkansas with children or with moms are all in the 50, 60, 70 participants, if not higher. And so it can become very large if you're trying to find very small differences and make them statistically significant. But that's a very good question, because in nutrition, we see a lot of pilot studies, a lot of studies that don't lead to statistically significant findings. So it's always important to have that. But think of your endpoint. What is it you want to prove? Is it a change in blood pressure? Is it a change in memory? Is it a change in appetite? Is it a change in gastric emptying? And then what is the variability of that endpoint? And then what are the group differences you would expect based, and if that variation plus the expected means allows you to do a power calculation, tells you how many a sample size needs to be.
Hello, I have a question.
Yes.
Thank you, Dr. Ferrucci, for your interesting presentation. I learned a lot from you. My question is, what do you think is the principal difference between wild grapes and grapes cultivated under standard conditions, I mean in greenhouse?
Would we see differences between grapes cultivated under normal, outdoors versus like grapes in the greenhouse, or like table grapes might be cultivated under cover and under control? You're going to see differences in the phenolic profile. The more the grapes are stressed, the higher the phenolic content is. It's the difference fundamentally between a table grape for fruit and a wine grape for production of wine, right? So wine grapes need to be stressed with heat, with drought, and with sunlight to make a very concentrated, high Brix, high sugar, high acid fruit that can be vinified well. So I would anticipate if you took the same grape variety and grew one like a table grape, i.e. you gave it a lot of water, you cut the root to a point where it would accumulate and grow a lot of pulp, versus growing it like under stressful conditions, that's the G by E, right? So you can have the same genetics, but you have the environmental effect. You're going to see differences in phenolic content, which means you might also see differences in other metabolites, which means you might see differences in bioavailability. So absolutely, I think if you talk about traditional cultivation versus let's say greenhouse or under cover, under irrigation, or if you have the same genotype that's growing wild, or at least some sort of, with grapes, it gets difficult because some of the wild varieties are crosses in and of themselves. It can be certainly different.
Thank you for your answer, Dr. Ferrucci.
Any question?
Good questions.
Some people is interested in the study of the polyphenols and interaction in the food matrix and the microbiota as well, I think so.
Yeah, I think maybe we should do this live next time. We'll bring more work. So I have to come down and visit you.
Okay, great.
But no, in all seriousness, if you have questions, I'm happy to send papers. I just appreciate the opportunity. This is always fun. And you have a great group of students there always. So.
Okay, and right now I think so that it's improving the students here, and they are participating more and more. And for this reason, we try to invite some professors from other universities to enrich this lecture in the graduate programs here.
Oh, that's great.
Mario.
It's good to see you. And
Okay.
Thank you, everyone.
Thank you. Mario Rodríguez, Mario, que puedes entregar el certificado, por favor.
Yes. One moment, please. Dr. Mario Ferrucci, thank you very much for your lecture. It's a pleasure for the Instituto Politécnico Nacional that you participate. And we, Dr. Luis is going to send the next certificate. This Instituto Politécnico Nacional, Centro de Desarrollo de Productos Bióticos, award the present certificate to Dr. Mario Ferrucci, Director, Arkansas Children's Nutrition Center, Department of Pediatrics, University of Arkansas for Medical Science, for his participation as a speaker with the lecture entitled Food Factors that Modify Absorption, Metabolism, and Potential Bioactivity of Phenolics, present virtually at the seminar of the doctoral program in Development of Biology Products, Yautepec, Morelos, 16 de mayo de 2022. Atentamente, la técnica al servicio de la patria. Thank you very much, Dr.
Gracias.
Mario Ferrucci.
Thank you, Mario. Mario, I will send the certificate your email this afternoon. Again, thanks a lot for your lecture and to accept this invitation. I appreciate it, Mario.
No, anytime. Sorry it took me a while to get the date down, but thank you.
Okay, thank you very much. I
Thank you.
hope that in a close future you stay here in Mexico, in Yautepec again. It
We'll
will
find
be
the time.
very kind.
I can need to invite you here. You would enjoy seeing what we're doing with children for sure.
I remember. Okay, thank you. Thank you very much,
Hasta la
everyone.
próxima.
Bye-bye.
Thank you.